Hello, I'm Caden Cooper, and for my 2023 State Science Day, my research is on comparative mercury levels in farm raised and wild caught fish. Prior to performing this experiment, I thought it'd be important to go over some important background information. I knew prior to researching I wanted to investigate in animal science or environmental science, as those are topics I previously researched last year. I found out how widely consumed fish were in the world and wanted to investigate any safety concerns for fish, as fish are the largest food commodity in the world. I also thought it was important to go over the process of mercury actually going into fish. So, carbon emissions that are released from factories have the ability to reach bodies of water such as oceans or seas, and those emissions have mercury in them. So when uh, when small organisms such as plankton get that into their body, those organisms are going to be eaten by smaller fish, and those smaller fish will be eaten by larger fish. So the process, the mercury concentration is likely to transfer all the way up to that larger fish. I wanted to see if trends such as that would align. I also wanted to examine how farm raised and wild caught fish differed in mercury concentration, as they don't have, as the fish have different uh, lifestyles and uh, food diets. My hypothesis stated that fish that are farm raised will have more mercury than wild caught fish due to their environmental surroundings. It should also be important to determine by how much fish farming affects store bought fish, along with determining which commonly consumed fish is more harmful and safest. Here's some more background information. Most mercury exposure to humans is from consuming fish and shellfish. Although the amount of mercury is not extremely high, consistent consumption of fish can lead to various health problems. What this means is if someone um, eats a fish with a relatively high mercury concentration, it's okay to have that fish every now and then, but if that fish is being eaten on a regular basis, that can cause health issues. Um, at the very worst, this can lead to mercury poisoning. That can affect the human brain, spinal cord, kidneys, and liver, as well as the ability to feel, see, and taste. This also affects specifically young children or babies, which can also affect their nervous system as well. At the very worst for adults, it can affect their brain and lead to kidney damage. I said families should know exactly how much mercury they are consuming when they select a fish to eat across the globe, as food standards aren't always the same no matter where you go. The purpose of this experiment is to compare the presence of mercury found in farm raised fish with the amount of mercury in wild caught seafood. Here's just a slide on my hypothesis and research question, as I previously went over. For the setup of this experiment, the materials you'll need is to purchase 10 to 14 fish at a local grocery store or fish market. You need to make sure these vary in species and determine whether they are wild caught or farm raised based on the packaging normally. Next thing you will need is a mercury test indicator, what I purchased uses the chemical phenyl 2-phenhydroxide. Next, you're going to need stainless steel trays as well as plastic cups, paper or plastic plate, pen or pencil, as well as a journal to collect data. For the procedure, select one of the fish that you have purchased and cut it open to reveal the skin. To use, next you're going to need the test kit. In order to use it, you're going to have to soak a cotton swab with rubbing alcohol and rub it against the fish for a minute. Allow the cotton swab to air dry. Mix the mercury indicator with half a cup of rubbing alcohol, which is also needed. You're then going to drop the solution onto your plate. Next, you're going to use the dried cotton swab and dip it into the solution. On the next slide, I provide a more detailed how-to for this mercury test kit. For the next step, you're going to plot the graphs for each of the fish's mercury percentage to compare them. For the test kit, it's able to determine the mercury concentration in fish down to the 0.1%. As stated previously, each fish was swabbed for 1 minute, and they were all the cotton swabs were lightly soaked in rubbing alcohol prior to the experimentation. Once all the swabs were collected, the indicator was mixed with the cotton swabs using a pipette. The swabs were left for 10 minutes, and then a cotton swab that was not swabbed with a fish was used to compare the darkness in each sample. Here are some photos that went along with the experiment. As you can see, the control variable is on the very right side in the top picture. What was used to determine whether the samples had more or less mercury was dependent on their if it became darker or lighter compared to the control. On the left is the incision of a porgy, which was one of the fish that was used. For my graphs and data, here's a list of each of the specimens. I used the numbers 1, 2, and 3 to determine whether each fish became darker, lighter, or had no change. For example, 1 means no change, 2 means lighter, and 3 means darker. Here's a table that corresponds with the graph that shows each of the fish's results. The next set of graphs compares the collected number of farm-raised fish and wild-caught fish and the results from the indicator. For the wild-caught graph, 1 specimen was found to have no change. 2 specimens were found to become lighter than the control, and 4 became darker. For the farm-raised, 1 specimen had the result of no change. 0 specimens became lighter, and 6 became darker. I also thought it was important to look at maybe if salt water or fresh water had anything to do with the results of mercury. For my freshwater graph, I found that 1 specimen had no change, 2 became lighter, and while 6 became darker. And I actually had the exact same corresponding data that goes with the salt water. For my conclusion, I said, the data collected appeared to have varying results from each category of specimens. The hypothesis of this experiment appeared to be supported in some cases but fell short in other instances. Due to the conditions of farm raised in their environment, it is predicted that most of them would have a higher mercury concentration. This was the case for the catfish, tilapia, rainbow trout, steelhead trout, orange king salmon, Cape sea scallop. The Acadian redfish, on the other hand, seemed to not change and be the same color as the control. While the exact details on why this fish did not match the trend are not known, it is a fact that these fish are a rather healthy type of fish to eat with minimal mercury amounts, which go against some of the fish in the category. The wild-caught specimens appeared to have more varying results than the prediction. Whiting and porgies became lighter than the control sample. The whiting and porgies became lighter than the control test sample, while the black bass had no change. The remaining specimens, the saltwater perch, swordfish, yellowfin tuna, and octopus, appeared to be darker than the control. It is important to note that how the fish was raised may not be the only conclusion on why fish had larger concentrated mercury than others. Fish that appear to have higher concentrations of mercury should be in moderation to prevent health issues. Sorry, I believe some of the lettering got deleted in this slideshow. For recommendations on how this experiment could have been improved, I said it would be more ideal if another round of samples were collected, as well as using a more precise indicator that can indicate parts per million with actual digit numbers. This would remove more room for error as well. Another thing I wish I could have done is been able to track down the specific water, a specific body of water each fish was from, like the actual river or ocean. That also would have been more ideal. And wraps up that wraps up my science presentation. I'd like to thank my parents and science teacher, Mr. Smith, for helping me along the way to this journey. And I believe that's all. I want to also thank all the previous judges that have judged me, and the current state science judges that will be judging this. Thank you for listening.